Okay, hi everybody. I am going to get started. It's just after 1 o'clock. So I wanted to just thank everybody for joining me here today and for coming even after rescheduling last week. So thank you for that. I was not up for presenting last week and you would have had to hear a lot of coughing. So thanks for your patience. And I'm hopeful that the session today is one that is helpful and informative and prompts a lot of discussion. And I'm interested to hear from everybody else as well. So let's get started. You guys should be familiar with these opening slides. This is the Systematic Review Workshop Series, and this is part 5 of 5. So you've made it kind of through to the end. And before we get started, I wanted to acknowledge where I'm coming from today in Winnipeg and on the University of Manitoba campuses. So the U of M campuses are located on the original lands of the Anishinaabe, the Ininiwa, the Anishinaabeg, the Dakota Oyate, and the Dene Sulene, and on the national homeland of the Red River Métis. And I respect the treaties that were made on these territories and acknowledge the harms and mistakes of the past. And I'm dedicating myself to move forward in partnership with Indigenous communities in the spirit of reconciliation and collaboration. So if you're joining from somewhere else today, besides Winnipeg, I encourage you to think about where you're joining from, and you're always welcome to share that in the chat as well, just to share where we're all coming from today and the lands that have been taken care of by other people for us for so long, and how we can better kind of acknowledge that and move forward. Excuse me. So today, as we are going to be talking about a variety of different tools, and our title is From ChatGPT to Covidence, Effective Use of Tools to Complete a Systematic Review. So before we kind of dive into the content, I just wanted to acknowledge again that I am one of several health sciences librarians, and for those of you who work or are affiliated with the Rady Faculty of Health Sciences, we're here to help you with any of your kind of knowledge synthesis questions, and you can reach us using that QR code and or the link to talk with us. And at the same time, I also recognize that we have people who are coming from other parts of the U of M community and even some kind of external people. So in particular, if you're coming from outside of the Rady Faculty of Health Sciences, whether that's engineering or anthropology or social work, we have librarians for each department in the university. And if you follow that link, you will see who the library for your area is. And if you have questions about systematic reviews, reaching out to them just to kind of get that conversation started can be a great first step. And the slides, I believe Janice has posted them a couple of times in the chat. They're also on the webpage for the systematic review kind of series. And so then you'll be able to click that link directly or go to the U of M homepage and access it that way. So a few housekeeping things. Everyone seems to know this by now, is just to keep your microphones off because we are recording the session. Same thing with video, and encourage you to ask questions via chat. And Sorry, I forgot to mention right at the top that my wonderful colleague Janice is joining us again today, and she will be monitoring the chat. So please feel free to send your questions to her. And if there's anything that I should cover, Janice, please let me know. And we actually don't have too many polls in this session today, but hopefully a lot of discussion. And at the end, I'm happy to kind of really open it up and have a discussion. And this session was set for an hour and a half, so that's how long we have. It's the first time we've kind of done it in this format, so it might not take that long, but I thought I would give us that extra time if needed. And for those of you who have attended all 5 sessions and are a graduate student, you can have this added to your experience record. Now that the sessions are complete, we will work on getting that done, and we'll contact you once it has been added to your record. If you don't hear anything kind of by mid to end of June, then you can feel free to contact me and I'll see what might have happened. And a reminder that all the slides and recordings are posted on our website. And I did send out to all registrants the slides and recordings from part 1 and 2. The slides and recording from part 3 and 4 should be up kind of today. So I hope to send them out tomorrow so you'll have access to them. And part 5, the recording will come out probably sometime next week. Okay, so the objectives for the class today, and some of them are, yeah, well, not probably what I would pick now that I've done all the content for the class. I might change them, but these are the ones that were there when you registered. So I'll leave them up there, but really it's about an exploration of tools to complete systematic reviews. And really, now that you've had kind of 4 sessions on the process, how can you help kind of streamline this process and make it easier for yourself and for the rest of your team? So those are some of the things that we're going to be looking at today. And in terms of the outline for the class, I'm going to have a kind of an introduction to tools with some caveats. And then these are the tools that we will be taking a look at. So if you haven't don't have the slides yet, I would encourage you to have them open just so that you can follow along with the links to kind of see how they work and see the interface. And I realized I didn't add the one of them is not on that full list, so I will correct that before sending out the slides, but it's listed later on. Okay, so what do we mean kind of when we talk about tools? And really, I want to emphasize something to think about throughout the entirety of the session today is that these tools are, the systematic reviews are complex themselves, and that the tools will not kind of replace the whole process of doing a systematic review, and they don't take the place of knowledge of the process and understanding it and expertise. But really what they are designed to do is to help make the process more efficient, so that if you kind of understand how to do, say, the search process, but are having trouble with translation from one database to another, is there a tool that can help you with that process? But if you don't understand kind of why you're building a search or why you need to have different vocabulary for different databases, you're still going to struggle. So really the tools are a supplement or kind of an add-on to the knowledge and expertise that you're kind of slowly building up through these sessions and through other readings and experiences with systematic reviews. And there are also many tools available. And the ones mentioned here are the ones that I personally use, or that my colleagues are used, or some that have been kind of strongly recommended as being helpful. And there are, I've tried to focus on tools that are either freely available, are available through the U of M, so the U of M library pays for them, or have kind of the ability for you to use for free for a little bit to kind of test it out. Because I could list tons of tools here, but all of them would have kind of costs associated with them, monthly charges or yearly charges. And that's not going to work for everybody. And it's certainly difficult for students, for researchers, for kind of early career researchers to allow for those costs. So that's why the tools that are listed here were the ones that were chosen. And there was a 2022 scoping review that attempted to kind of identify all the different software tools that were out there to assist in the systematic review process. So this is just kind of a diagram or a figure that shows you the different tools that were identified for each kind of step of the systematic review process. So if you really want to dive into the different steps, you can see, but really what I'm hoping to show you is that there are a lot of different tools out there, and that you want to find and use the ones that work best for you and kind of meet your needs. So if you don't have a lot of funds, well, what are some of the open source or tools that are available through the library that you can make use of? And then maybe you can explore other tools later on. So there are a lot of tools out there. And I'm not familiar with all of these. There's too many, and whether tools are available or not kind of year to year changes. There was one that I wanted to show in this session, but unfortunately it hasn't been working for the past couple of weeks. So, you know, things change, technology changes, and access to some of these tools will change over time as well. And so this is just software tools, this one here. But I also, in this session, what I've also included are some tools that would kind of fall more under the heading of either kind of AI or more machine learning and automation tools. And we didn't always talk about these in the past, but I think everybody is much more aware of kind of the capability of some of these tools and we want to kind of think about and talk about how can they help you streamline the systematic review process. So some of these tools that are more kind of machine learning or generative AI kind of tools, they can help potentially mitigate bias that might come from the people who are on your research team. Again, not intentionally, but just say how when we talk about the process of screening, which is when you decide whether an article gets included in your systematic review or not, human screeners can have bias. So can we do some kind of automation or use machine learning to help kind of change that process? And they can also help us to uncover patterns or connections that a single human or even a team of humans can't do on their own, just for the sheer scale of some of these projects. So probably with these types of tools, more than some of the other software tools, kind of the more traditional tools, you always want to very carefully evaluate the outputs. So yes, use them, explore them, try them out, see what works, but really pay close attention to what it's suggesting and don't kind of just take what they are suggesting you do kind of at face value with no kind of appraisal or analysis or kind of a deeper dive. And what is really nice about some of these tools is the ability to minimize some of the repetitive tasks. And there are repetitive tasks in systematic reviews. Screening is a big one. Another one that kind of we've moved past, or I've moved past a lot of doing these days, is the deduplication process. So they can be really beneficial, but you still want to think carefully about those tools. And this is from that same 2022 scoping review. These were machine learning tools that can assist in the systematic review process. So you can see, if you compare to this one, there are tools kind of for each step of the process that have been identified. But when you look at this one, there's fewer tools, but they are kind of focused on specific stages of the systematic review process. I would say some of these have changed, and there are going to be kind of tools that will assist you in the other kind of steps, but this is an ongoing process. And I would say too, this was 2022. So if you think of kind of how scoping reviews or systematic reviews are done, this data was probably collected in 2021 at the latest. So think about how much this field has changed since 2021, 2022. So there would be, I think many of the tools that I'm talking about today aren't even listed here. So kind of there's just lots out there. Okay, so now let's talk about tools. So I've emphasized again that these are the ones we've chosen that we use or that I use and that are kind of more readily accessible. And when I first organized this session, I kind of did it how those scoping reviews was organized, as in these tools, this is helpful when you're at the first stage, and then so on and so forth. But what I really discovered is that a lot of the tools can be used in different ways and at different stages of the systematic review process. So instead, what I'm going to do is go through the tools and then identify kind of at the end what I think they're most appropriate kind of to be used for. And I did attempt to kind of go from start to finish, but there will be some overlap. So again, these are some links out to every tool that I'm going to be talking about today. Again, if you just want them kind of in one place. So let's start with the biggie, ChatGPT or any of the kind of chat bots that are out there that have really changed kind of a lot of things so fast. So these ChatGPT along with some of the other models like Gemini or Perplexity. I've listed them here, Copilot or Claude. What they do, right, is they have ingested massive amounts of data, and then they essentially kind of interpret, and you can have a kind of a human, like a natural conversation with them. And there are free and paid versions. And so I chose, although I've listed kind of similar tools, and I myself tend to use Gemini the most, I chose ChatGPT because I think this is the one that people are most familiar with using, and even kind of are most familiar with even thinking or talking about. And so at first you might wonder, well, how can I use this to do a systematic review? And I can say that some people are already using it probably in ways that I would not advise, almost letting ChatGPT take over the whole or large parts of the review. But where I find it kind of particularly helpful is when you are starting your project and you want to think about how do you go through, you kind of are at the brainstorming stage. So what I've done throughout this whole session is kind of worked off of one kind of very broad area of interest, which is the use of exercise and its impact on smoking cessation rates. So if I was interested broadly in this topic and I wanted to know more and I kind of wanted to start developing my search strategy, ChatGPT can be really helpful for that. So what I've done here is I've linked out to a very kind of very simple conversation that I had with ChatGPT to, let me just bring it up here, to kind of get us started. So Janice, let me know if at any time you can't see what I'm talking about, but you can see here, I've just put in at the very top, and I'm using the free version of ChatGPT here. I'm about to start a research project related to exercise and smoking cessation. What are some important things to consider? So at the top, ChatGPT is going to walk me through kind of how to do a research project and how to do what I want to consider, all of those kinds of things. And these are important to think about, especially if research is new to you. And then, but I want to get kind of deeper. I'm interested in systematic review. So I want to say, okay, but I want to do a systematic review. So if I want to build a comprehensive search strategy related to exercise, I'm just focusing on one concept right now, using medical subject headings. And then I'm asking it, which subject headings should I use? And so what they're saying here, and is they will give you, ChatGPT will provide what it thinks are appropriate MeSH or medical subject headings for you to use. So I put in MeSH because that's what I use in the health sciences. But if you are in a different discipline, you can ask for subject headings that are related to your field of study, or you can ask for just keywords. So keywords can be used in any database. They aren't subject specific. So again, if you're coming from, the environmental sciences, you might just want to use keywords based on the databases that you would use. So you would ask ChatGPT for some help with brainstorming. So at the very first step is, okay, well, here are some terms for exercise that I might want to think about. And here are some subject terms for smoking cessation. And in the past, I probably would have generated these search terms by looking at articles. So I would have looked through relevant articles and tried to see what subject terms they were using. But now it's already, the top or the terms are being presented to me and just saving me time. And then what ChatGPT goes on to do next is say, okay, well, here's how you can combine them. And it's giving me examples of combining them using Boolean operators, which are those and or that we talked about earlier in the session. And what they've done is actually put the code right here. So the code continues on. You can see it goes quite far. It is put it into what I could copy and paste right into PubMed. And it goes on for the individual concepts and then combining them together. So I could copy this and paste it directly into PubMed for that search. You can see it actually does it right here for you. It then also gives you some suggestions. Here's what you might want to do. You might want to add on additional concepts, additional filters, and they reinforce the idea that searching is an iterative process and that you might want to make adjustments kind of as you go. But then I say, okay, but what if I want to use a different database instead of PubMed? And so what it will do is it translates it essentially from how you search for exercise in PubMed to how you would search it in Ovid Medline, a different database, and provides you that translation right there. And it does it for both of the concepts. And then it does the same thing. It tells you this is how you would build, this is what you would copy and paste into Ovid Medline, and this is what you would copy and paste for those two concepts and then put them together. And then there's your final search. So again, this is just coming, and it's giving me additional stuff that I might want to read. But again, from a very simple prompt, essentially very simple, this is what it's building for me kind of right, right off the start. And then what I've asked in this question here is I've said, okay, well, I've got my search in Ovid Medline. What about if I want to use Ovid Embase, a different database? How would I translate that? And again, they give me some more discussion points for me to consider, and then they do the translation for me. And in this case, there was a lot of overlap. I would want to check these because, and I should have done it to ensure that. And this is what I mean about checking the work. So I would definitely want to check it. I wouldn't just run it in Embase and assume everything is perfect, but it kind of gets things started. And that kind of, again, it's just giving me tips, and it's a conversation, and I could make modifications based on the feedback I was getting. But from very simple prompts, kind of getting a great place to start for building and developing my search in Ovid Medline and in Embase or in PubMed if I wanted to do so. And I will note that some of the things that it suggests are not totally accurate. So for example, it suggests, exercise and then specific types of exercise, but it doesn't really tell you that if you search exercise like this, if you explode exercise, you're already capturing all of these. So you actually don't need to do those three because exercise would already have captured it. So it's little things like that, that complexity that isn't kind of quite at that level yet. But again, a great place to start and brainstorm. And the second thing I wanted to highlight is this is very simple prompting, but you can have much more complex prompting. And in the slides, I link out to where I have taken these prompts from. I did not develop them. But this is a more kind of elaborate or complex one. And you can see is what it's trying to do is build kind of a multi-step conversation with ChatGPT on developing a systematic review kind of search. So it says things like follow my instructions precisely, don't explain or elaborate, only respond with exactly what I request. And what it's saying is, given the following statement and text from a study, identify 50 terms or phrases that are relevant. And then of those terms, we're going to do some more work. So you can see here is they copy and pasted an article title and an abstract. And then what ChatGPT does is identify 50 words related to that. You can see here they are, 50 terms. And then the next step is for each list, classified into 3 categories relating to health conditions, terms relating to treatment, and terms relating to study design. So what's happening is the terms are being generated. Now the terms are being classified kind of by concept. And then the next stage, let me scroll down. Then for each one of these, it's basically prompting Pub to build a more complex PubMed search based on what is found. So that's what's happening here. And I'm not going to kind of go through this, but now it's being kind of translated into MeSH. So this is a much more kind of complex search that is being generated, and it's basing it off of kind of a single abstract that has been provided, instead of kind of me just saying, oh, this is the broad topic that I'm interested in. So again, this just shows you the potential for how you can use ChatGPT depending on how familiar you are with kind of building prompts, which are the questions that you ask of it. And I've already started using it just to help me kind of generate terms and ideas for how I might want to kind of get started on a new project. So that's not to say those are the only ways you can use ChatGPT, but just kind of the first couple of ways that kind of occur to me, especially in the search term development, I found it particularly helpful. Okay, and yes, I listed as well the ones that I find similar and, oh, pardon me, go back. There we go. Where I found it most helpful. And if people have already started using ChatGPT for their reviews, I'm interested, feel free to put it in the chat if you found that it was helpful, wasn't helpful. I'm interested to hear about people's experiences. Okay, so then that's ChatGPT. Let's kind of take it in a sort of a very different direction, which is once you've got your search and now you want to start writing your protocol, and you want to in particular write the methods section of your protocol. So there is a kind of project or a group that has developed something called the Systematic Review Accelerator. And then within it, they have a whole bunch of different tools. And we're going to look at a few of them today. One of them is called Methods Wizard. So what this allows you to do is kind of pre-fill information in the program, and it will then write your methods section that you can then edit, but it kind of really just takes a lot of the guesswork, especially if you've never written the methods for a systematic review before. So let's open this up. Actually, I have it, I think, already open here. Okay, so I have, you can create, I should have been logged in already. I think I was, but it logged me out. Okay, accounts are free to create. Hold on. I'm going to bring it in over here. Kind of, it takes a second to load. The reviews are, pardon me, the accounts are free to create. Everything is free, but now we're just going to wait for it to kind of load. I guess it signed me out on this one. Let me see if I can go back. Okay, yeah, here it is. Okay, so I've already started pre-filling some of the information just to kind of move the process along. But what you can enter in is, and all of this information is based on a study that's already been published. This is not my information. But I put in, okay, well, here's the title of my study. Here's the year that it's published. At this point, you can add who are the people who are working on the project. Is there anybody who helped you? And you can upload your research plan, which is kind of like the steps or the stages. And then what you really dive into are the more specific parts of your methodology. So you can write an introduction, would kind of be the literature review. So basically, why are you doing this project in the first place? What information gap are you hoping to fill? And then this is where I find it particularly helpful. So let me make it a bit bigger so people can see that. Okay. So what it's asking for me, what it's asking for right at the top here is, what are your eligibility criteria? So you can say here, what's the population, the people, the problem that you're interested in looking at, and whether that is to be included or excluded. So I have put in here, and this is actually an older one. That's why I'm not doing the smoking one, but bicyclists of all ages who have crashed or fallen while riding a bicycle. So those are the people I'm interested in looking at. And then I haven't put any exclusion ones, but I could. So I could say, well, I don't want any bicyclists younger than 18 and older than 65, for example. And then what are your interventions? And in this case, it is the use of any type of bicycle helmet, including hard shell, thin shell, or no shell helmets. And I could exclude at this point, say, other specific kinds of helmets. If you're wearing a hockey helmet, I'm not interested in what happens if you get into a bike crash. And the comparator is if you are wearing no helmet. So I have typed that in there, wearing no helmet. And what are the outcomes that I'm interested in looking at? So again, for a systematic review or for a lot of different types of reviews, you'll have usually a primary outcome that you're interested in looking at, and then some secondary outcomes. But you can have multiple primary, it kind of varies. But in this case, I've listed what are the outcomes I'm interested in. I want to know about head injuries. I want to know about brain injuries. I want to know about serious brain injuries. And I'm interested in facial fractures or lacerations. I have put my setting. In this case, I didn't leave anything, but I could. And then I would also indicate what study types I want to include. And then if you scroll down right here is output. And if you click on that, you can see that they have generated the text for your protocol or for your method. So some of this you will definitely need to edit it and play around with things, but you can see they have typed up, okay, here we're using all these different study types. They've used the information about the population I'm interested in. They've looked at the interventions. They've looked at the outcomes. They put the participants. They've kind of broken it all down. So it's not that any of this is particularly difficult to do, but especially if you've never written this before, this can be a huge time saver, not even just thinking about what format should I use, but having it all broken down like that. So that is just one part of the methods wizard. There's also the search strategy. So you can put in who developed your search strategy, who ran it, what components did you use when you created your search strategy, who did your search, and was it peer reviewed? Were there any restrictions, any language restrictions, etc.? What were the additional parts of your search that you did? So did you contact experts, all of that? Your output, again, formatted for you. You'll have to make some changes, and because some of it I didn't kind of completely fill out, but again, you can just copy and paste that into your protocol. And there are one of these kind of for each one. So there's a more extensive one related to your actual search strategies, including the databases where you searched, how you did the screening, how many authors independently screened results, and how many for both stages, and that kind of thing, how disagreements were resolved, and whether you adhered to kind of the reporting guidelines for Prisma. So again, you're just kind of toggling a button and then getting an output. In this case, I haven't put in a lot, so I don't have a lot of output text. And then you also have forms for data extraction, for risk of bias assessment, measurement of effect, unit of analysis, and missing data. So I typically haven't done these ones, but all the other ones, excuse me, and then just copy and paste, take all those kind of all those outputs, put them into a Word document and edit it, and then you're essentially done your protocol. Just excuse me for one sec. Okay, so I hope that would help and kind of get you started as you go forward with your projects, and to encourage you to write and register protocols. I think I was muted there. So that was methods, the methods wizard. So where I thought that this would be most helpful was obviously, sorry. Setting up your search, drafting your protocol, and writing your paper. Okay, so the next one, which is kind of a new one for many people, is a product called Elicit. So Elicit is one of the tools that I'm talking about today that, sorry, I thought I put my cold behind me, but that is not available for free and isn't available through the university library. So it works on a credit system. So if kind of you're interested in what I show you today, Elicit is one of the ones that you kind of get a certain number of credits, and you get free credits at first, so you can use it to kind of try it out. And if you think it would be helpful, what you can do is, once you're about to start your project, start like a 1-month trial, pay the $20-ish for 1 month and really use it well during that 1-month period. And then if you need to end your subscription, then you can go ahead or do that. But what...
Mê-Linh?
Yes, Jen.
I was just going to ask, did you want to switch back to your PowerPoint?
Oh, yeah, I do.
Yeah. So we can see the slide for how Elicit is spelt and what it can do for us. Thank you.
Yes. for sure. Okay, showing now. I'm assuming, just let me know if it's not.
Yeah, it's great. Thank you. Thank you very much.
Thanks, Janice. Okay, so Elicit combines large language models with scholarly sources. So Elicit, as well as some of these similar tools here, so Consensus, Cite AI, and the other ones here, they all pull from different data sets. And some of them, most of them are pulling from Semantic Scholar, which is actually an open source set of scholarly papers, but some of them will also sign agreements with publishers. So for example, I haven't listed it here, I did Elicit, but Cite AI is one that I've used kind of more frequently. So Cite AI, this one right here, has signed an agreement with specific publishers. So they are paying publishers for access to their scholarly papers. And the reason that makes a difference is if all of these sources are essentially pulling from the same data sets of articles, they all don't have access necessarily to articles that are behind paywalls, that are only available through subscription. So they're just showing you, say, open access articles, or they're just showing you kind of the abstracts of the articles. So that's why they don't have everything. So don't think that any of these has access to all of the scholarly material that is out there, and that if you look kind of in one, well, that's enough. It's not kind of, it's not correct, because that's not how they're getting their information. But what they can be really helpful for is showing you the linkages between papers, and also those kind of connections. But what I like about Elicit in particular is how it helps me extract data from papers that have been identified by Elicit, or that I have uploaded into Elicit as the papers that I'm interested in, and I want help kind of extracting data. So let me show you what that looks like. So here, and I did print screenshots, and again, I preloaded this because this takes some time to do, because it's looking through all this content. So again, I did my very basic kind of, I'm interested in doing a systematic review on the role of exercise and smoking cessation. So let me make it a bit bigger. So that's what I prompted Elicit with. So then what it has done is written me a summary right below, and you can indicate to Elicit how many papers you want it to summarize, essentially. So I was on the free account, so it only did 4. If you pay to upgrade, you can get up to 8. So it talks about the articles, and it's actually mentioning several systematic reviews, but it's kind of giving you a very high level overview of a field, to kind of give you some ideas. And then what it can do next is it will identify some articles that it thinks it's important for you to consider for this topic. So you can see here, there's an article from 2000, from the journal Addiction, from 2022, from 2021, 2012, 2022. And I actually don't, some of these tools, it's really hard to know why it's making the decisions that it did. It's not totally clear why these specific ones were chosen. I'm assuming that somewhere in the algorithm, there's like a relevance ranking, and that these ones were chosen because maybe they're high level reviews, or they're more recent, or it's what Elicit has access to. So that's kind of not totally clear. But then what I've done is you can see that running along here are specific columns. So this is kind of, if you've ever looked closely at a systematic review, is what kind of, this is the data extraction table, essentially. And this is what kind of a lot of authors spend a lot of time reading these articles closely, pulling out this information, and then they do kind of some kind of synthesis or analysis. But what I've indicated are the columns of interest to me is I want a summary. I want to know what the main findings of each paper are. I want to know what their specific methodology is. I want to know what the intervention, independent variables, and how many participants. And you can see kind of tucked away on the right-hand corner, I have ran out of credits for Elicit, but you can see that there are a whole array of other columns that you can add in, and you can also add your own column. So you can indicate more specifically what you want Elicit to, what information you want it to extract from the PDFs that are in there. So I know I indicated that you, I guess I should be cautious about this, but Elicit doesn't have access to everything. You can upload your own PDFs to Elicit and it will extract the data. So then if you had access to articles through the library subscription, then it could pull data from those ones. So especially if you had a Plus account. And then I'm just going to scroll down. So it actually it's just showing me the top, I think this is 7 or 8 articles, but you can add more, and you can go on to kind of subsequent steps. But this is what I have found really helpful is the data extraction. That's how I've been using or have used Elicit in the past. So I would recommend this one. Try to create an account, do some basic searching, try it out, and then see if you want to, if it will be helpful enough and you want to kind of motivate yourself to pay for the account and try it out yourself. And the other ones I listed here, the one that I've used the most is Cite AI. I like it as well, but I didn't use it for this one. But I like that it has access to additional content and just the interface, which looks very similar to Elicit. So I would say it's helpful for getting familiar with your topic and doing some data extraction as well. And then I just put some screenshots in case Elicit wasn't working, but it's just what I showed you kind of just right now, but a little bit more detail on additional columns that you can add. Okay. So that one's definitely more of the kind of generative AI tool. This one is kind of flipping back again to more of a software tool, but has been around for a little bit longer. But so again, you've got if you used, say, Elicit to help you think about a field, and now you want to actually, and you've got some key articles, but you want to identify more important keywords that you need to use when you're developing your search strategy, you can use the word frequency analyzer. So what this allows you to do is take a whole library of articles and kind of dump them into the tool. And then what it will do is output common words or phrases that appear within that library. So you do need to have articles, like seed articles, identified already to use this tool, but you can do it with one. That's not particularly helpful for one. But even if you kind of do some basic searches and get 5 to 10 articles, you can upload that here to get some ideas. So again, this is a freely available tool from the Systematic Review Accelerator program. So let me open it here. Oh, no, this one here. Okay. So this one actually moves pretty fast. So I'm actually just going to upload it right here. So you just choose a library. It has to be in a specific format. And then I'm just going to upload it. And it does a and I didn't, I'll clear this in a second and show you. But it does a very fast frequency analysis. So it finds the word smoking. So there were 75 articles in that library. It finds the word smoking 59 times, exercise 55 times. So, your most common terms, those are pretty easy for you to kind of figure out. But as you kind of get into less common terms that might still be really helpful and maybe would identify some of the articles that your search wasn't finding, these can be really helpful. And you can also tell it kind of which kinds of terms you aren't interested in as well, but it will show you essentially how many times the article, oops, let me go up all the way to the top, kind of how many times it's been found, where it was found. Usually if a word appears in the title or the abstract, and it's what you're interested in, that would obviously indicate that something is likely to be of more relevance to you and you might want to consider this more. than if a word was only found kind of one time in the abstract versus 2 times in the title, as an example. And let me just close this and show you. Before you kind of analyze anything, you can tell it, what if you wanted to ignore common terms, so like the, and, etc. You wanted to ignore numbers, duplicates, all those kinds of things. You can also tell it how many words can be in a phrase. So I don't know if you noticed, it had the words randomized and controlled side by side, but I could move that up to 3. So anytime the word randomized controlled trial appears, it would be given that single term instead of 2 separate terms. So you can definitely play around with it as well. And it's, I like to kind of use it as a double check. Are my keywords, have I captured the important ones? And freely available, so always nice. Okay, flip back. Here, and there's instructions on how to use it. And this one, again, helpful for kind of developing your search strategy. And I put a screenshot there to kind of show you what that looks like. Okay, this one, it's been around for a while, but I actually kind of like it better. And for the non-health sciences people, I'm sorry, but it is a tool that identifies medical subject headings from already identified studies. So again, if you found, say, 3 or 5, and you want those articles to be included, you can pull out what's called the PubMed identifier, the PMID, and I've kind of indicated it here where it shows up. It's a 8-digit number unique to each article, and you can load it into the tool. And I'll show you what that looks like. Okay, so here I've just chosen, again, I did it already, but it happens very fast. You can see there are 5 different articles that are loaded up here. Let me make it a bit bigger. And then what it does is it breaks it out. So here it puts the title, it puts the author, not as helpful, but where I find it really helpful are the MeSH headings. So it shows you the MeSH headings for every single article, and it's alphabetical, and it's grouped by kind of by letter. But all of these articles that I chose all had something to do with exercise and smoking. But what you can see is how variable the subject heading is. So for example, this older article from 2013 has the subject heading of smoking, but the newer one from 2024 has cigarette smoking. Again, another one, you can also see that this one here has smoking and smoking cessation. This one has just smoking again. This one has just smoking. So you can see kind of the differences between articles, and you can also see what if there is kind of a common subject heading that you need to include no matter what. So for example, exercise, 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 exercise. But where it's really helpful is if there is an article that is helpful, but for some reason it's not showing up in your final set of articles, and you're like, what's wrong with my search? Why aren't I finding it? Throwing it into something like this can be really helpful, because you can see, okay, for example, this second one here, the second column, it does not have exercise as one of its subject headings. So it has walking instead. So if for whatever reason I had not included walking in my original search development, I had only included exercise, but I know that I want to find this article, I would go back, edit my search, and add in walking. So it's really helpful for identifying articles if you don't know why you missed them. Because what's happened is I've worked on a number of reviews where I submit kind of my articles to the team, and then they'll come back to me and say, well, you didn't find this one. Why isn't this one in there? And so you have to kind of do this analysis to figure out what went wrong. Because if you missed this one article, are there other ones that you may have missed using those same subject headings? And you can also, just so you know, it's kind of listed right here. I think I have a screenshot of it. But if you're in PubMed, it has a little bookmarklet essentially called Analyze MeSH. You can be in PubMed and you can click that and it'll automatically kind of pre-fill this as well, so that you don't have to. This is what it looks like. Usually is you copy and paste your PMIDs in here and you tell it kind of what you're looking for and hit go. So just a small kind of simple tool that can actually be, actually save you a ton of time. Okay, yeah, so I showed here just in this screenshot of how smoking can be captured differently in these different articles. And that you can add that bookmarklet. Okay, so this is actually one I didn't know about. It's called MeSHmate. Again, a MeSH one. Apparently, we really like tools to help with MeSH in the health sciences. But what it does, anyone who's tried sometimes to find MeSH, it can be a little bit tricky. And I would say that this one is not perfect, but it's a really kind of very simple little tool that can be helpful. So I have it listed here as well. Okay, so I think I had a couple ones that I wanted to show. So for the first one, let me put in smoking. Okay, so I type in a keyword, I type in smoking, and then what it will do is it will identify MeSH terms that I might want to consider for my search. So here you could see, okay, maybe I want smoking cessation, I want cigarette smoking because I saw it from that other one. And then down here on the bottom, you can copy those terms with like copy and then paste, being able to paste them into PubMed, and you can use and or to kind of build that search. So I find it helpful for, or I just learned about it, but what I found since I learned about it is I find it helpful just to kind of give you an idea of terms that are out there. Some that are kind of pretty standard. Likely I could, figure out most of these just kind of from kind of brainstorming. But it will also show you how terminology changes. So if I put in indigenous, it will show me, for example, health services, indigenous peoples, and will also show kind of the older, outdated terms that we don't use, that it's not language that we use anymore, but some of these databases kind of still, unfortunately, do use them. So you have to use them in your search, even though they can be offensive. So it's showing that language is changing, but some of these databases are unfortunately slow to change. And so how can you kind of incorporate both kind of sets of terminology to be as broad as possible? What I did find though is that, and I couldn't find this in the help, is that I'm finding I'm not sure when it's updated. So I just put in COVID-19, which definitely has a subject heading, and it is not listed. So I'm not sure how current it is. So I wouldn't trust it, again, as my only source, but as a very quick kind of brainstorming tool that's easier than looking through the PubMed MeSH database. I really liked it for those reasons. Okay, so again, helpful for search development. The next one is called Systematic Review Accelerator, the Search Polygot. And so this is a tool to help with the process of search translation. So if you put in your search for one database, it will translate it, the search syntax for different databases. I want to emphasize here, you still need to understand how the databases work. And you also, it doesn't include subject headings. It will only include the keyword search part of your search. So what that looks like in practice, again, I've got it loaded here. Okay, so basically what you do is you copy and paste your search. So I'm pasting a search that I did. And this one is not the smoking and exercise one. I wanted to include one that had a lot of keyword searching with what are called the adjacency, right? So this one was about sex and biological variables for drug development. So I would say things like, I want the word sex within, say, 5 words of biological variable, or the word sex within 2 words of gender, which itself is within 2 words of the word based. So just a different search to show you that complexity. So I have pasted in this search, And then if I scroll down, I can expand and it will show me what it looks like in a different search. So if I click on, say, CINAHL, which is a nursing and allied health database, it will give me this part. It's confusing. It's showing you a subject heading, but it hasn't actually translated it. It just changed this part, which is the syntax part. So you still have to find out in CINAHL what is the subject heading for gender identity. But where it's helpful is for the keyword part. So here you can see it took my, let me scroll up. It took this sex within 5 words of biological variable, and it translated it to the word sex in, it's kind of this teal title or abstract or subject heading. And then it uses N5 biological variable and the variations thereof. So that can save you a lot of time, especially as if you have trouble remembering, I do, what the different syntaxes are in the different databases. It will also tell you if it has trouble with a particular search, so you would want to take a closer look. And if you scroll down, you can see kind of goes quite long, but it has some of the major databases. Even it does have health sciences ones, but also like Scopus isn't necessarily Web of Science. Those would be kind of broad sciences and actually have a lot of social sciences material as well. So that is the Search Polygot, again, part of this Search Accelerator suite of tools. And helpful for search translation, obviously. And then another one that's still in beta from the same kind of company, or not company, it's an organization, is called TERA. So this one allows you to identify additional relevant articles that you might not have captured. This one, I don't love it yet, but I think it has some kind of potential. So it's here. And what you do is you can add a whole library of articles, or you can just add PMIDs. So you can see here what it's doing. I've put in those 6 PMIDs, and it's listed. It says here, these are the articles you've used in your search, and now I want it to search. And it's going to link out to articles. So we'll see right at the top here, this is that article that also Elicit found by that usher. So exercise interventions for smoking cessation. And it indicates how relevant it thinks it is to your search. So actually one is of more relevance than say 10, but one is the most relevant. And the more articles you upload or you share, the more article suggestions it will give you. So this can be helpful again for identifying, just kind of starting your topic. Like we talked about seed articles. How can you figure out seed articles if you haven't built your search? Well, if you have 1 or 2 good articles, put them in here, and it will then give you some more articles that you can quickly look at the title and abstracts to see, oh, do I want my final search to include this? How would then I kind of build my search around that? So it gets quite lengthy. I think this was down to, I think I would wanted to show me almost like 250 articles, but the more articles you add in, the more it will show you. Oh, now here it is. 338, and I had 6 that I put in as kind of like my seed articles. So again, getting started, figuring out how you're going to develop your search, and also then you could use it as well. I'd be a little bit more hesitant to do this, but you could also use it to identify articles you may have missed in your original search. But it means that you might have to kind of scroll through a lot of things. So I'd be cautious about that, but it has potential there. Okay, this is just a very small one that I just want people to be aware of. It's called PrdictTER. And so if your advisor says to you, well, you can do this systematic review and you can do it in your 6-week research block or over the summer, maybe you can give them some information on how long it actually takes to complete systematic reviews. So I put it in here, PrdictTER, and oh, hold on, let me reload it. So what you can do is you can input information on how much time you anticipate spending on each phase of your systematic review. So are you only going to spend 5 days project planning? Maybe you've got the methods wizard. So you're only going to spend 2 days writing your protocol. How much time does the lead author have to spend on the project? Are you doing this full time, or are you also doing this while, working full time and doing something else as well? So how much time do you have of your kind of day to devote to this? How many databases do you plan to search? Do you plan to search gray literature? How are you going to, you can then put in how many articles you have identified, for example, because that will really change how much time you have to spend screening. So if I say there's 4,000 articles found, well, that means you have to spend about 4 days screening. If I say I found 10,000, my screening time is going to go up. So they estimate about a day for every 1,000 articles to screen. And you can kind of do that for each stage. You can input the information that you have. And again, it's an estimate, right? It's a prediction. It's not perfect, but just to kind of raise awareness for both people working on the team and maybe people thinking of getting involved with the team about how much time it can take, especially depending on how much time you have to devote to these different stages of a systematic review. Okay, so Abstrackr is one of the ones that I had, there were some problems, I had some problems with it over the last week or so. So I think I did, yes, I included a screenshot just to kind of show the process of how it should work. We'll take a look and see if it's actually working this afternoon. But it is a free, it's online tool, and it is designed to help in the screening process. So this is one of the most time-consuming steps of one of the most time-consuming steps of a systematic review. So for those of you who might not be familiar, what happens is in a systematic review, once you have your, say, final set of articles from your databases, so let's say you have 5,000 articles, at least 2 people on your research team need to review all 5,000 of those articles first, just the title and abstract, to decide if they should be included or not. And then after title abstract, you need to do a full text review to see whether it should be included or not based on your inclusion and exclusion criteria. So as you can imagine, it takes a fair chunk of time to do, and is one of the reasons that a lot of kind of projects I think kind of fall apart, is people just don't have the time to devote to screen the articles to include or not include. And so companies have been working on how to streamline this process. So I've listed again some other similar products that do kind of the same thing, but Abstrackr is free, and it also incorporates what's called natural language processing and active machine learning. So what that means is you can toggle this kind of off and on. But let's say you have 5,000 articles to screen. What it will do is, if you turn kind of this kind of active learning component on, is as you screen, it is learning from you. So it is learning what articles you think are relevant and include versus what you don't exclude. And the more articles essentially that you train it on, the better it gets at predicting what will be included and what will not be included. So as it learns from you, then what it starts doing is it starts pushing the articles that it thinks are more relevant up to the top for you to screen. So you are screening what it believes are the more relevant articles first. And that can be really helpful because if you're screening all the ones that are relevant first, and then suddenly you go through, let's say 100 or 1,000 where none of them are relevant, none of them are included, well, that tells you that probably the rest of them aren't going to be relevant either. So you can kind of go a little bit faster. It doesn't mean you cannot screen them, but you can kind of go faster. It's a different level, attention level, than if you're screening 5,000 articles and you don't know within those 5,000 articles, where are the 25 articles that are going to be relevant. Like the level of kind of attentiveness and detail that you need to do, if the relevant ones can be anywhere, are different than if you know they're more likely to kind of be top loaded for you to screen right at the top. So some of these have incorporated it in different ways, kind of in levels, but that's basically how they work. And I think, let's see, no, not, it's still not, it's kind of a little bit, it's a little bit sluggish. So let me go back, I'll show you what it looks like from this screenshot. Okay, so this is a screenshot, and what it is you're doing kind of your first round of screening. So here's a title, here's an abstract, and what you can do is you can set tags. So you can indicate to the system, okay, if you come across these sets of words, highlight them in green, because those are things I'm looking for. But things in red are things that I don't want. And I don't know what they've tagged here as blue. So you can use some different kind of color coding that then helps you to be able to quickly look to see whether something should be included or not included. And then you would just go through and check the checkbox to say whether to include or not include, and kind of go through your list. And at the same time, other screeners can be going through their lists as well. So it can really speed up that process. So I haven't used Abstrackr myself because I only do a moderate amount of screening, never kind of, let's say 5,000 or more, but I can see it being really helpful to streamline that process. So think about this if you know you're going to be screening. And yeah, the other one, one of the reasons I've listed it is because it is free. So ASR Review, I really like the interface of it, but it does have a cost associated with it. Ryan, you can use it. You can definitely create a free account, and I can't remember how many projects you get with the free account, but it uses machine learning as well, this active machine learning. And I want to say you can have at least 5 projects before you have to pay, but don't quote me on that. But they have a very nice interface as well, especially for a tool that you can access for free. Okay, obviously, yes, helpful screening. Okay, and then I wanted to talk about Scopus. This isn't one that you would think, we talked about it in databases. You could be like, well, why are you including it as a tool? And it's because it has a feature that can be very helpful closer to the end of your project that you might not be aware of, which is it is somewhat, sorry, if there's another librarian and then they can tell me for sure. If it is listed that you have to do what's called citation checking for your included studies. So what that means is, say at the end, you've got 10 studies in your systematic review. What you would do is from those 10 studies, you would do a citation kind of check for the articles that have cited it since it came out and the articles that it itself cited, so backwards and forwards, to make sure you haven't missed anything. You don't want to do that on every article because that's a lot of information to look for. But once you have your included studies, you can very kind of quickly see that. And Scopus is definitely, well, I find one of the easier ones to use, and you do have access to it through the library. So I've put it here what this looks like. Okay, so I'm going to do this one here. So I'm going to pretend that this article, Exercise Interventions for Smoking Cessations, is one of my articles that I want to include. So if it shows up, what I would do is I would enter the article into Scopus, and then I would, on the right-hand side, you can see it's got 16 citations. You would want to use these, and you would quickly look at which 16 articles have cited it since it came out in that one. I can't remember what year that was, 2019. So you would do a quick skim through that and make sure that if that you have all of these articles, or you've looked at all of them, and decide whether you would want to include them or not. And you would also open the article itself up, which you can do by clicking on the title. And then you can read, if you scroll down, you can look at the articles that it cited. So again, a quick kind of skim. And that's why I say you only want to do it for the articles that you have decided to include, because it can take a can take a bit of time. So you want to just really focus on the articles you know are really relevant for your study. So that's just a different way to use kind of one part of Scopus that you might not have been aware of. Okay, and I think the last one, let me just double check, is so some people say Covidence, some people say Covidence. We have done a full hour and a half on Covidence. So obviously what I touch on here is not covering all of the features of it. It is an online product. It has a cost, but we have a license here at the U of M library. So it's free for the U of M community. You just have to create your account the first time through the U of M, and then you can then access it kind of just covidence.com, I think, going forward. So what it is really nice for is you can import your results from different databases, and it will deduplicate your results. Deduplication used to take a really long time. I'm not saying that Covidence is perfect at it, but if it can save you, you know, hours and hours of effort, and it's kind of good enough, that can kind of be good. You can also then look at all of the articles, and basically you can screen right in Covidence for deciding whether to include or exclude. You can also build your data extraction tables right in Covidence, and it will auto-populate a Prisma. So it's helpful at lots of different stages of the systematic review process. So I'm going to show you my account so you can, oh, actually, I think I forgot to log into this one. So I'm just going to quickly log in so you can kind of see the different parts of it. So I wanted to show you a few different ones. Okay, so this is just a test one, bike helmet laws, but this is what it looks like. So you can see here, this is your project. So this would be your systematic review. What you would do is you would import your studies from your different databases. So you did your search in Medline, you did your search in Scopus, and then it will remove any duplicates. You can go and look at the duplicates to make sure they truly are duplicates up here. You can read those details. And then what it does is it moves it to the title and abstract screening stage. So it's telling me, Mê-Linh, you've still got to screen 481 more articles. And out of a total of, I think I've screened 50 so far. So here's what it looks like. If I hit continue, so this would be the very first step of the screening process. So title abstract. So if I was screening for this, I would be thinking about, okay, I'm interested in, you know, bike helmets and safety. And I would, right from reading this title and abstract, I would make the decision right here about whether to include it or not include it, or maybe I'm not sure, I want to have some future discussion. So I would go yes. I'm just going to do this quick. No. Children, I don't want children in my study, so I'm going to say no. And no, I don't want kids again. Okay, lots of childhood ones. No, global news highlights. Okay, but maybe this one here. Okay, so I'm going to go through those almost 500 articles. At the same time, I have another person who is doing this same process, looking at the same articles. And we're looking at the same articles to ensure that we have agreement. So if we have conflict, which hasn't happened, it would show here, you would say you have 3 articles to kind of resolve. And then we would have to decide, we would either meet together and decide, or we would have a third person screen and decide whether something should be included or excluded. So it's like a double check, right? About whether to include something or not include something. Once all the articles go through the title and abstract screening stage, they go to where you screen them for full text. So at this point, you are reading the full text of the article to decide whether it meets your inclusion or exclusion criteria. So you can see here, the full text article is uploaded. I would have to read through it. I would have to know what I was looking for in the article. And then I would have to say whether to include or exclude. And if I exclude, I have to say why I'm excluding it. You can't just say exclude, because you have to be very explicit in what is basically what is wrong with the article, not why doesn't it meet your inclusion criteria, and part that there. So I find the interface for this really nice, very streamlined, and easy for multiple team members to work together to go through the screening process. It also works, I'm going to go back to another one of my reviews so you can kind of see this. So I also say you are involved in a review that kind of takes years and years to do. So I'm involved in a review that looks at the use of cannabis for children in clinical trials. And we run a search every single year. So I'm just showing you kind of the history of those searches. So every year I run new searches for the team to look at. And by uploading them, you can see it's going back in time, 2023, 2023, 2021. So by keeping it all within a single project within Covidence means that they don't have to worry that they are looking at material they've already reviewed. It's kind of keeping updated for each year as new information comes in. And I'm sure we've mentioned it probably in some of the other classes, is that reviews take a long time. So you're probably going to have to run your searches multiple times and look through multiple sets of articles. So this can really help with that. And then I also wanted to show you what it looks like to do a data extraction table. So this is a different review that I'm working on. We ended up with 48 studies in our final kind of set of included articles. So if I click on that, I want to show you what this looks like. So here's an article that we included. And if you click on this, it will show you what information we pulled out from the article. And it's got a I should have said it this way, set it up already, but you can just very easily kind of quickly skim the article and fill this out to allow for kind of easy data extraction. And you can build this specific data extraction kind of table right within Covidence. So you can tell it, I'm interested in the title of the article. I want to know the study design. I want to know this intervention. I want to know this. I'm interested in this dosage of medicine. I'm interested that it took place in the Arctic. Like all of those kinds of things you can say here in your data extraction template. So when you're going through the review process, you can very quickly pull that information out. And then you can download it into Excel or CSV for kind of additional analysis. And also, let me show you, where did it go? When I talked about how it will generate a Prisma flowchart for you. So here you can see, here is the Prisma flowchart generated. I haven't done anything for this. It shows you how many studies were found at the beginning. It will show you where those studies came from, how many duplicates were removed, how many articles were screened, assessed for eligibility. It'll tell you why. Again, it's counting what you kind of input and then how many final studies you have. So again, you can do this manually, or you can have a tool like Covidence kind of create it for you automatically. Okay. So I think I put in some screenshots just in case. Covidence was a little bit slow today, but it worked nice and fast. Just the different parts showing you kind of what you would see. And that is the last tool that I have. I did list a few additional resources, especially for those who might be interested more in the kind of machine learning, automated side of systematic reviews. What kind of things to consider, where can you be kept updated about what tools might be out there. But really, I would say you need to work always, if you're new to systematic reviews, work on the fundamentals first, and then kind of try and find tools to help support that. I also have put in the evaluation, and I'm happy to answer any questions, or if people want to share their experiences, I'm happy to hear that as well.
I was going to say, Mê-Linh, that was great. There was a lot of information there, and I'm glad there's a recording and some slides. And I think, it really excited people when they saw different things, because anybody who's got a little bit of experience doing a review might have kind of glommed onto something that said, oh, that would really help, right? And I
Yeah.
know that for myself, I don't know all the software. So often what I do is I do a search in Scopus of the type of software that I'm interested. So if somebody says to me, oh, maybe try that distiller software, I can do a search in Scopus or Google Scholar, and I can put in the type of software, the name or the URL, the website for the software, and put in maybe the term software, and then put in systematic review or scoping review. And I can find how other people have used it, if they've used it. Sometimes it doesn't really matter what the subject of the review is, but sometimes I can just get an idea if other people are using it and perhaps how they're using it. So I found that really helpful, and getting lots of thank yous in the chat. So thank you.
But that's a great idea, Janice. Yeah, seeing how other people are using those tools. So I always find just sharing what people are doing is one of the most helpful ways. I learn a lot from my colleagues and from the students and I work with. So yeah, I will update the website and provide the slides and the recording. And really, I know it's a lot of information, but I wanted it to just be, even if you just remember that this tool exists, even maybe not how to access it or use it specifically, but then are, yeah, willing to kind of go explore it and talk to us if you have questions about it. We're happy to chat. Okay, well, if there aren't any more questions, I will end the session. And thank you for everybody who participated in the series. And hopefully it was beneficial. And again, just encourage you to provide feedback. And yeah, have a great rest of your day. And I'll be sending emails out soon. Bye, everybody.